today I'm going to be talking about KombOver, which is our work on characterizing dynamics and shifts in microbiome. We will mostly focus on host-associated microbiomes because this is a healthcare conference, but this is relatively agnostic work, so if any of you are looking at something like soil or marine microbiome, this can still apply. Now, the talk itself will contain some fairly technical bits, so the slides will be up afterwards and I'll be around. I'll try to focus on relatively high-level picture because I realize that a lot of people in the audience might not be super proficient in genomics, metagenomics, and so on. So what's the motivation behind our work? Well, we want to establish the connections between host microbiota, so microbes that live inside all of us, and the disease phenotypes. This is obviously relevant for gut-related diseases because that's a big harbor of microbiome in our bodies, but also can be relevant for some skin conditions and so on. We also want to scale what we're doing to really large data sets because, again, as everything in metagenomics right now, we're looking at terabytes upon terabytes upon terabytes of publicly available data. And this is essentially an extension of prior work we did, so we have some foundational work that shows that indeed those methods can pull out very useful and interesting thing in case of fecal matter transplant patients and so on, but the prior work was not as scalable. So in general, what do we want from tools that analyze metagenomes? I would classify this in two categories, things like low processing times and low processing costs, like running on a laptop. This is the question of scalability at large, but also something that provides us with granular information at the patient level, because the dream of everybody is personalized medicine would tailor everything to you, not just sort of one-fits-all solution. And we want to work with biomarkers that are interpretable, something that a clinician can look at and say something meaningful, not just a bunch of numbers. So I would generally refer to this as richness of the information provided. So to the first point of scalability, we can think about metagenomic sample as a collection of these circular, in this case, genomes of our microbes. We put it onto a sequencer machine, something like an Illumina, and we get the short strings out. So these are our sequencing reads. Now, in general, we will get millions of reads from a single sample. So there's a lot of data, and we want to compact it without losing any information. So this is where de Bruijn graphs come in. Again, I will kind of not really go into details of what exactly de Bruijn graphs are, but the way you can think about them is you have some string information, you're trying to compact it and represent it as compactly as possible without losing any information you need for downstream analysis. So in this case, they have a property that if something was a full genome, then that same string of characters will be preserved in a compacted de Bruijn graph. So this is kind of what you want to retain here, that these graphs are essentially lossless compression of our read data. Now, however, as this lossless compression is great, it's also somewhat devoid of additional information. In particular, Illumina reads have this nice property that there's a pair of reads you get from every single molecule. And what that means is we know that for every pair of reads, they came from the same genome. So how can we leverage that information? We leverage that information in our tool by constructing what we call a hybrid unity graph, where in addition to the edges that we get from the data compaction in a de Bruijn graph, we also augment it with some additional edges, shown in color here, that correspond to information contained in the paired end reads. So we basically link up things based on paired end information. You can think about this as starting with compressed, sparse graph and making it much more dense. Not that crazy dense. These graphs are still very much sparse in the context of things like social networks, but in context of genomics, these graphs get more dense than a typical situation. So now that we have a graph, and this is probably something where a lot of AI folks will be like, aha, I get where this is going. We have a graph. What do we want to do? We want to locate important nodes. We want to find what is the driver, what is the important part of this graph. So there are a couple of algorithms to do that, and we'll touch a little bit on machine learning later on, but obviously there's betweenist centrality. Everybody loves betweenist centrality. Google really used to love betweenist centrality, but it has one huge caveat: it's slow. So you can see the complexity here is number of vertices cubed, and we're talking about graphs that can have millions, tens of millions, and in really bad cases, hundreds of millions of vertices. So cubic runtime that's not scalable. However, that was previously implemented in a tool called Bambus 2, and it does tend to pull out biologically relevant things. It just doesn't scale to the data sets we have right now. Well, we can do approximate betweenist centrality. That is linear runtime, much nicer, also implemented before, metacarval. However, here comes the problem of approximate betweenist centrality. It starts being fuzzy, and it's actually not as good at pulling out significant or biologically interesting parts of the graph. It's a bit more noisy. And also, I will say that at least from our experience, it doesn't really do as well in terms of pulling out things associated with any phenotype. So that leads us to the idea of core A score. Core A just stands for coreness anomaly. And behind all of this is the K-core decomposition algorithm. So something that is linear in the runtime and is also precise. So this is not an approximate algorithm. You can run it linearly in the size of graph and get exact coreness value for each node. And now by comparing coreness value of each node to its degree value, we can find outliers, the anomalous nodes about which we will talk in a second. So what do we mean by this? I will not go through the technical primer on what coreness of a node means, but the easiest way to think about it is high coreness means high interconnectedness. Low coreness means I generally don't have that many connections. I fall off the graph very fast. But it's not exactly the same thing as degree because, again, this node falls off the graph quickly, yet has relatively high degree, which is what we want to capture with the anomaly score. So something that tells us this node doesn't seem to fit the connectivity pattern you generally expect in a graph. There's something odd about it. Or it's really well connected, but compared to overall degree distribution, its degree is still relatively small. So these are the nodes we're going to be hunting for. Now, a quick reminder, behind every node is in fact a biological sequence called unitig. So what we really get by hunting for those anomalous nodes is we get fragments of genomes that have interesting connectivity patterns. They typically would be something that is either particularly enriched, so high copy number gene, or it can be something that participates in horizontal gene transfer because it appears in a lot of things. So again, this is kind of the main goal of hunting for those nodes. So I was talking about scalability first, and in terms of scalability, we do offer quite an improvement on our previous iteration of the code. So in terms of wall clock time, we go down about 7-fold. Some data sets at the top, those are like sort of your standard big-ish analysis of metagenome. We have 200 samples, about 2.5 terabytes of data. Single sample we can process in about 6 minutes. So basically, this is relatively good, I would assume. If you have a patient, you can run, like processing for 6 minutes is definitely much shorter than running Illumina for 24 hours. So in general, processing gets relatively fast. We also have some data of running this on human genome. This was not really built for human genome. Human genomes have ridiculously more complex repeat architecture. Really what we're showing here is you can get a very complex graph. The tool still scales to that. But what does that tell to us in terms of actual pathology? So we took a look at this chronic fatigue syndrome data set previously published by Xiong et al. In 2023. Really nice data set, very well curated metadata. Part of the reason why we picked the data set because we could really sort of take what was published before as the ground truth and see whether we can pull out similar biomarkers. So we've started by just looking at general distributional properties. There definitely seems that people with the chronic fatigue syndrome have less unitics, so that basically means we could assemble less of the bacterial genomes, generally correlates with what was observed in literature previously. People with chronic fatigue syndrome do have mild or severe, in case of the short-term fatigue syndrome, dysbiosis. So this kind of like high-level proxy, but what really is interesting to me is digging into like sort of richness part of this. So this is a case of 3 matched samples. So unlike some other studies, in this study, each patient donated 1 sample, so we don't have time series. But I did manage to locate patients with exactly same age, sex, kind of other phenotypic descriptors that fall into 3 categories. So the healthy control, the short-term chronic fatigue syndrome, and the long-term chronic fatigue syndrome. So what do we see? The profile that we're looking at, the overall graph, is tiny. The degrees are low. The cornices are low in the short-term chronic fatigue syndrome patient. Again, this matches exactly with the dysbiosis hypothesis. We also see the somewhat recovered trend in the long term. So in general, for chronic fatigue syndrome, it has been observed that short-term patients have pronounced dysbiosis, while long-term patients exhibit partial recovery, yet the richness of the microbial community never fully recovers. So it's kind of like this in-between modified state that they arrive at. And one of the interesting things that somebody who knows a lot more about microbes than I do can do here is take these high anomaly score things and start looking at those nodes directly. So again, each node is a sequence. We did some very high-level analysis of these data. In most cases, these highly anomalous unitics that we pull out, specifically in the short-term chronic fatigue syndrome patients, they do correspond to some of the bacteria that have been shown to be enriched in those patients. So again, this is kind of a way to get at those annotations, at those sort of questions much more directly. And one of the sort of sidebar discussions here would be, well, why not just classify taxonomically everything in the sample? Like, we can also get at this or something like that. Yes, but at the same time, by having longer sequences, so unitics are longer sequences, you actually are able to add functional information. You can functionally annotate. You can try to find not only what species, but also what protein this is associated with. The second part is, when we're talking about highly anomalous unitics, we're talking about a sort of compaction of our data, a next level compaction of our data. So, for example, the number of highly anomalous unitics, which we define as 3 standard deviations above the mean for the anomaly score, is about 1% of all unitics in the sample. So if you think about having 1 million strings with annotation information, no doctor in the world wants to look at 1 million things. Now, if I'm talking about 10 things, somebody might actually take a look and make a meaningful comment on that. So that's an important part here. We also took a look at some other summary statistics on this graph, just looking whether indeed the graph structure varies with the phenotype. Very much does so, and we very clearly see that there are different shifts. However, again, this is more sort of high-level observational data, and we really want to dig into the details later on. We also compared the richness, well, in this case, actually alpha diversity. Again, the similar pattern talked about before. And some of the future work that we want to look into is, so far all we're doing with this graph is predefined. We are thinking about what's a good metric, what's a good thing to measure. But one of the promises of AI, especially in terms of graph neural nets, is if we have a graph, can we learn what features of the graph are important? Can we also, while doing that, find a good idea of what is the null model of a healthy graph. Can we understand what is the base so that we can better start quantifying what is the anomaly? So these are some of the things that we're planning to look into in our next steps. And again, we want to think that maybe we can define not just the difference between healthy and diseased, but what kind of disease as well. Again, this requires, first and foremost, scaling to large data sets. I would like to acknowledge Trangen Lab, in particular Dr. Michael Nude, who was helping with some of the statistical analysis, as well as offered a lot of helpful critique. I would also like to thank Ken Kennedy Institute, in part as a recipient of last year's Andrew Ladd Memorial Excellence in Computer Science Fellowship, NIH and NSF, which partly funded the work. And we have the GitHub for anybody interested in trying out the tool. And the preliminary version of this manuscript will appear in Proceedings of Pacific Symposium in Biocomputing.
We have time for a quick question.
All really cool. Thinking about like what cool questions you can address with graphs. Are you looking at like quantification and variation at all, or is it mostly like what other summary statistics do you think you could use these graphs for?
Oh, absolutely. So in general, some of the things that we always wanted to look into but struggle due to computational scaling is looking for things like graphlets and submotifs, generally searching for any submotif bigger than like 5-node thing is nearly impossible. But those are definitely one of the things we would want to look at and are looking at in some other projects, in part because when you have something like a horizontal gene transfer event occurring, you actually can describe it with relatively small graph, like what a biologist would expect to happen given that case. Now, there are some attempts from other labs' work to look at things like counting triangles or counting K4s in the graph. Some of it is interesting, some of it is a little bit kind of not always as informative. So I would say we're definitely at least looking into motif mining, but we're also thinking, when thinking about motif mining, we're kind of pulled between two directions of letting the data tell the story or asking a biologist first to provide a good prior. So, yeah.
Cool, very cool. Yeah, because I'm like, I guess I'm aware of like alignment-free methods and alignment methods, and they always require like an actual like bacterial reference in like database essentially. And so like you assembling these graphs, it's opens up new avenues.
Oh yeah, and it's very nice that you bring up the reference-based methods, because one of the beauties of this is it all happens database-free. So this is kind of like one of the plagues of the field. It's less problematic in human health because we actually surprisingly understand human gut bacteria well, at least in terms of who's there, but it's absolutely horrendous in environmental samples, so yeah.
Thanks.
Thank you.